So indeed, we heard already from Chris a bit of the method, so what comes before, and now we'll get into the maybe more interesting part. So you do something with a goal, and now that goal is to eventually diagnose patients. So how do we do this result interpretation to reach that? So what is needed is this integrative analysis in which we analyze the RNA not alone, because the RNA alone is very difficult to reach a diagnosis just with it. So we analyze it with the DNA and with the clinical reports. But that's not the only thing that I mean by integrative, and I find it very important to also combine together the bioinformatician part with the medical part and with the experimentalists. And working together, it's way faster and more precise. We'll get a diagnostics from experience. So this has been done already throughout the years since 2017 from the pioneering studies that we had here in Munich and at the same time at the Broad Institute in the US. And basically all they do this to detect this aberrant expression, splicing monolithic expression, and combined with the information from DNA, either from whole exome, whole genome sequencing, we have had some diagnosis uplift or from 8 to 35% in different, taking different tissues, blood, muscle, and skin usually, and in different rare disorders. And now in this year, we have had a actually great uplift in number of studies, and now it's really becoming worldwide. It's not just exclusive to US, Canada, and Germany as before, but now we have studies from Hong Kong, Netherlands, Australia, and UK. So it's definitely getting popularity, this approach. And basically, we have two different ways to detecting outliers. One is what we call the RNA first, in which all expressed genes and junctions are tested. So now we're talking about the tens or hundreds of thousands of features that are tested, which makes multiple testing correction quite harsh. While many times in diagnostics, in rare disease diagnostics, we have a set of candidate genes that we're interested. For example, if we test even all omnium genes, we're just in the 5,000, or some patients we already know the disease they most likely have, so then that can restrict to the hundreds. If we test already genes that have that are known to have already high impact, we're just 10, 20s, or other candidates we could be really 2, 3. So we reduce a lot the search space. We have adapted our methods to do that, both Fraser and Outrider. And indeed, for example, in the blood cohort from SOFAR-D, which is one of the biggest ones that we have, we see that indeed it's complementary to focus on certain genes than with respect to testing all, but of course, testing all will give even more results in total. So we have worked throughout the years with different data sets from different tissues, from different disorders, for example, ALS, mitochondrial, maybe the one that we have studied the most here in Munich, the UDN, which is the Undiagnosed Disease Network, mostly of US, and lately, since last year, with the SOFAR-D as well. And you see it's usually hundreds of samples, and our methods, both Outrider and Fraser, they return just a handful. Maybe Outrider is around 5 outliers per sample, and Fraser is usually less than 20. So it's not a lot to inspect. Especially if you add these to phenotypic and genetic information, you end up with very few, 2, 3 cases to inspect per sample. So why do we do this? So why detecting even aberrant gene expression? So we have this gene that is very much reduced in a specific sample with respect to a population, because it can be indicative of variants. It can be indicative of variants even before the gene, in enhancer promoters, in the UTRs. It can be indicative of large deletions. This has happened many times, that the deletions were missed by a standard whole exome sequencing analysis, and through finding aberrant expression, we can find them. Or it can be due to intronic variants that they activated a new this exon contain a premature stop count, and that led to aberrant expression. So I'll present now some cases. I think five in total, real life cases that happened to patients where RNA was directly useful. For example, this one, a patient from Japan, from this Chiba University Hospital that was a couple years ago, suffers from a mitochondrial disorder. Whole exome sequencing revealed a heterozygous missense, probably damaging variant in NFU, which is a well-studied or well-characterized mitochondrial disease gene. Here is the variant, the missense variant that was found by whole exome sequencing. Now this gene is recessive, so that means we need variants in both alleles. So this, even though it's very promising, is not enough to diagnose. Then what we find with RNA it was a 50% reduced expression. And a missense variant usually doesn't cause reduced expression. Moreover, we found that the missense variant here detected by whole exome sequencing was monoallelic expressed. That means that there's something going on in the second allele that is causing full loss of function. So what we did is we performed whole genome sequencing, and indeed after performing it, we found quite a large deletion, more than 4,000 base pairs, encompassing a full exon. And that solved, now we have these compound heterozygous variants, and that solved the case. Second case I wanted to show was another variant in the promoter. So this was a patient already with neonatal disease of leukodystrophy. And whole exome sequencing did not report any candidate variant for this sample. While then RNA revealed for this sample only 2 genes that are aberrantly underexpressed, and the highest one is being UFM1. And UFM1 is a well-characterized gene that is known to cause exactly that disease, the one that the patient had. And then that led to West reanalysis, and indeed there was found a 3 base pair deletion in the promoter region. And because it's so high, it was discarded depending on the place and the workflow. Some centers tend to discard variants, even if they're found by whole exome sequencing, tend to discount variants up to certain 100 or base pairs up or downstreams or intronic. So that's why this variant was completely discarded, even though it was detected. And because this same variant was already reported as pathogenic since 2017, the case was solved. So yeah, those were 2 specific samples, but we have many of those. Here in the x-axis, I'm showing the expression fold change with respect to the cohort. So this is the average in 1, 50% means is usually indicative of a heterozygous loss of function variant, while a higher reduction is usually indicative of biallelic or homozygous loss of function variants. And indeed, we show in all of these examples, these are the disease causal genes, that's the outlier gene. The gray dots are the background, the whole cohort. And we usually see that in case of heterozygous, we have, it's a bit high, maybe 30 to 40% reduction in the RNA. And at some point, it becomes stronger when we have a biallelic loss of function variants. And that's the previous one was direct, right? You have a loss of function variant in the gene, and that leads directly to underexpression in that same gene. But we have found other very interesting cases of consequences, so of downstream consequences. So it's not the same gene where you have the mutation that is necessarily the one that is aberrantly expressed. So what do I mean here? For example, this case, the causal gene was this gene LRPPRC. And that gene is known to regulate the stability of mature mitochondrial transcripts. And that the disease causal variant was an in-frame deletion, and we see the expression is just slightly reduced, is somewhere there. So it's definitely not an outlier, because and it makes sense because it's just an in-frame deletion, so not out of frame. And indeed, we see all these mitochondrial DNA genes underexpressed because this gene is messed up, and that it disturbs the stability of those transcripts. The same with this other case, LIC3 is a gene known to cause the disease called mitochondrial DNA depletion syndrome. And again, we see many mitochondrial encoded genes underexpressed, including LIC3. LIC3 is also there because it has one stop variant. So in this case, the stop creates the loss of function, is also underexpressed, so unlike the LRPPRC, which is just an in-frame deletion. So 2 super interesting cases of how through the consequence, we were able to find and or confirm the disease causal effects of these genes. So that was aberrant expression. So now going to aberrant splicing, we saw already with Chris that is a way harder problem because it can take multiple forms. So aberrant expression is usually just the gene is up or down regulated, but with splicing, it can take all these forms because of different variants. For example, a variant here in the acceptor site can cause exon skipping. An exonic one can cause exon truncation. One in the direct splice site can cause exon elongation. One in the intron, super common ones, can activate an exon, or can cause a full intron retention. So again, I'll give some examples. So this one from Sofardi from Epicare, the ERN. It's a female patient that suffers basically from epilepsy. That's what this ERN investigates, epilepsy. DNA through no candidate variants, I think, after whole exome sequencing. And then through RNA, in this case in blood, we found 2 exons skipped. So here you can see these two exons, they're skipped in 50% of the reads. So sometimes they are taken, but not all the time. So it's very common to do sashimi plots when investigated, but then you're able to see only one case. And what if this alternative splicing is common? And that's why we detect aberrant splicing, not just alternative, in this cohort of more than 250 samples. And then we see that usually the observed Jacquard index is around 100% for all the cohorts, except for this sample of interest for both the donor and the acceptor side. In both donors, so from this side and from this side, then 100 except for the sample of interest, in which is reduced basically 50%. And when it's reduced 50%, it's indicative of something going wrong in just one of the two alleles. So through DNA reanalysis, we found indeed a heterozygous deletion here comprising these two exons. This was confirmed by qPCR, and it was known to, or discovered that it was transmitted from the father, but it's fine because incomplete penetrance has been reported for this gene, which is a dominant one. Another case is this of a patient that suffers from acute liver failure. Whole exome sequencing didn't reveal anything, so just whole genome sequencing was performed, and that also didn't reveal anything interesting. Maybe the most interesting case was a synonymous variant in this gene twink, because it's a gene known to cause mitochondrial DNA depletion in the brain and the liver, and so it fitted the phenotype of the patient quite well. However, it'll be very difficult to diagnose a patient with a synonymous variant. So RNA-seq was performed, and then we saw that exactly on the spot where the synonymous variant is, the exon got truncated. It's homozygous, so we see it's full exon truncation. And that doesn't happen in other samples. So this is just a control. We don't see this truncated part both in the coverage, in the full coverage, or in this sashimi plot. So this variant is activated here, a new splice site, and that leads to an out-of-frame exon truncation, which also caused aberrant expression. So here we see the consequence that was not really expected from this synonymous variant, and that's why it could not have been solved just by the DNA analysis, even though it did capture the variant. Finally, the last case I will show, Chris already touched a bit on this gene TIMDC1, where a new exon was created with respect to other samples where we only see weak splicing. And why do I touch again? Because, not just it's an example of exon creation. So since then, this gene was already published in 2017, since the original publication of Kremer. But other unrelated individuals have been published since with the same variant. And that motivated the group to try to design some treatment. So it's not common to see genetic treatments yet. Basically, what they did is using these oligonucleotides, a correction of this intronic TIMDC1 variant that was reported 5-6 years ago. And the idea is that you fix the missplicing, and because of that, in the cells, and because of that, you can cure the patient or get a treatment for the patient. But not all is perfect with RNA-seq. It's not 100% effective. We saw from the studies that it just uplifts by 8 to 35%. And why is that? Well, first of all, not all genes are expressed. So here we see all protein coding and OMIM genes. Depending on which tissue you take, you get around 60-70% of the genes that are expressed. And that means you cannot test the rest 30-40%. You're completely blind to that. Moreover, we are usually restricted to these clinically accessible tissues, which are blood, muscle, and skin. It's basically impossible to test the brain, which would be probably the most interesting one for example, neurological disorders, liver, heart, and so on. We are simply restricted to those three. Now, even if the gene is expressed, the second problem is that there's these isoforms that are expressed in a tissue-specific manner. For example, this gene, TRPC6, we see that these 3 exons are expressed in the lung, but that's not the case in the brain. Finally, because we are detecting outliers statistically, we need sufficiently high coverage. So if the mean read counts are too low, we simply cannot, we're not able to recover outliers. We see at some point we recover 100% of them if the mean is quite high. But if it's very low, we simply cannot. So if not only the gene is not expressed, but if it's lowly expressed, we cannot detect at least underexpression outliers. So how to overcome this? one way is by predicting. So here we developed this method a couple of years ago, was finally published this year in the lab. The idea is to predict RNA splicing in any human tissue. So there's two or even more well-established tools that are in diagnostic in the labs. Usually SpliceAI is the most common one, but none of those are tissue specific. So what we design is these so-called splice maps, where we say if there's a variant here in the intron, but there's no exon here, like in the brain, so in the stomach and in the heart should be fine, but because there's an exon here, it will impact the brain, but not the other two organs. That was the main reasoning. So and we were able to develop this using GTEx, so more than 16,000 samples to check the variants and the splicing. The first thing we did is compare a SpliceAI and ME splice, and as ground truth, we used the splicing outliers called using Fraser on all the examples, all 16,000 of them, and we see that the precision and the recall are quite low. So by using this, removing underexpressed genes, taking into account and use splice sites, plus taking into account weak splice sites, plus using the scaling law of splicing that says basically the splicing is always between 0 and 100%, but it matters a lot, which is already the reference. So basically the average across the population, it matters where you are at the start. So, and that influences how much the can happen. So using all of that, we were able to uplift the precision of MSplice up to 3-fold. So we are here with this method that we call AB splice DNA, way higher curve than the rest, and now it's tissue specific, and it was published a couple of months ago, and it made it to the cover of Nature Genetics. So that's a way to do it in case we want to predict, for example, what will be the consequence in the brain. We cannot test the brain in the patient, but at least we can try to predict using GTEx. So the conclusions of this part, first of all, diagnostics should be interdisciplinary, not just restricted to certain specific fields, but like it's better to work in a team, on my experience. Then RNA-seq can and has increased diagnostic rates by two different ways. The first is validating candidate genes or variants. Then the second avenue is to suggest new genes to be inspected, usually genes that were missed by whatever reason, through whole exome sequencing analysis. Then it has had an 8 to 35% uplift in diagnostic rate. It varies. It's a large variation. Why is this? We think it depends on how you recruited your patients. Sometimes they're already candidate variants, sometimes not. Also, the disease entity and the probe tissue might have a say there, but this is more or less the standard rates of diagnostic. Then we have seen that fibroblasts is the accessible tissue with the most expressed genes with respect to blood and muscle. So one recommendation could be that samples are collected upon the first visit, and that fibroblasts are quite good because they allow for follow-ups experiments, unlike blood. Then we developed AB splice, which is tissue-specific RNA splicing prediction, and that can help overcome not probing the desired tissue. And finally, there's potential treatments via splice switching antisense oligonucleotides that are still in the make. Future directions, so where can this technology lead us to, or what are the immediate and short-term and medium-term directions to take? So all of this presentation has been based on what is now called short-read RNA-seq. But long-read RNA-seq is emerging, and it has the potential to quantify isoforms more precisely. So here the question is more like how cost-effective it is, or it will be in the near future. And when that happens, we'll have to adapt all our tools that we have showed. We don't know if they will work as they are for this technology. Then very interesting would be now to integrate with other omics, for example, proteomics or metabolomics, also both statistically and biologically see the impact, how they can be modeled together, for example, or use them together to conclude, to give more strength to a diagnosis. Then also, most of the cases I showed, we saw that it was whole exome sequencing that was failing, but now we have whole genome sequencing that is also becoming cheaper and therefore more accessible. And whole genome enables the study of complex variation. So it's not just big deletions, but also inversions, translocations, duplications, short tandem repeats, and so on. So The question here will be, can RNA-seq capture their consequence? So we'll need to have cases with those different variants, and then coupled with RNA to be able to investigate how good RNA-seq performs there as well. Then regarding the methods, so again, all the cohorts that we have inspected, it's usually in the hundreds of samples. So are the methods ready to cope now that I guess in the very near future, for example, with Genomics England, they already have thousands of samples. In Germany, it will also escalate. So are the methods ready to cope with that, or do they need to be adapted as well? And then for DNA sequencing, there's usually these platforms that allow you to inspect RNA-seq events. But for RNA, there's not such thing. So I believe a browsable platform will be very much useful for this purpose. So with that, I'd like to thank also from Chris' side, well, we both work and studied in the Technical University of Munich, so especially Julian, our PI, and many other colleagues that we've worked together throughout years, developing the algorithms, different algorithms that I presented here, also from the handholds from the lab of Holger Prokic, where we started studying these problems since, I think, 2015. And then we worked together with them in all these mitochondrial disease cohorts. From other institutes, I also showed examples from, in this case, from Barcelona, Japan, and UK. From SolveRD as well, I showed one example from Epicare, but now that's where we have the latest data sets that we're working very hard to get some interesting solved cases. And of course, all the patients, including in the studies and their families and the different funding.